Marilyn, the floor is yours.
Okay, great. So thank you very much for inviting me today. The organizers have asked me to speak about how to design a phase 3 clinical trial of a herbal medicine using the example of FACA. So I worked together with the team in Burkina and David Petrov, who I think is on the call here today, and Sika in preparing a proposal for a phase 3 trial of FACA a few years ago. So I'm just going to take you through really some principles of how we did that hopefully will be useful for you. So the first thing to think about is, why do you want to do a clinical trial? And there could be several reasons for doing a clinical trial. Maybe you want to prove that a herbal medicine works, or maybe you want to prove that it's safe, or maybe you want to prove that it doesn't work. Some people may want to do that, or to prove that it's dangerous, or to test which medicine is best, or to test how safe and effective it is compared to another medicine, or compared to a placebo. I think ultimately what I'm interested in as a doctor is to be able to advise patients on the safest and most effective treatment for them. But I think it's important to set out by thinking about, what question are you actually trying to answer? And in terms of herbal medicines, I think the first step is to think about, do you have sufficient justification to do a phase 3 trial? A phase 3 trial is not a small undertaking. It's big, it's expensive, it's time consuming. And so you have to consider whether someone is even going to want to fund this. So FACA is a good example because, like Sylvain has just shown us, there's been a lot of research on it over a period of several decades. So that's a good example of a plant in which there is good justification for doing a phase 3 trial. So you have to consider, is there a convincing signal of effectiveness? Presumably, if you're proposing to do a phase 3 trial, no one has done one before, so you won't have proof that the plant works from a randomized controlled trial, but there could be some convincing evidence from ethnobotanical studies, from preclinical studies, or small-scale clinical trials, exactly like the ones which Sylvain has just shown us. Obviously, you want to know, is the product safe? You want to have good evidence on lack of toxicity, not lack of effectiveness. I apologize for that. That's a typo. Will the product address an unmet need for patients or for health services? There's no point in really doing a clinical trial on something for which a good treatment already exists, which is already available, affordable to everybody. Again, FACA is a good example of this. There is clearly an unmet need for patients with sickle cell anemia, and there's no good conventional treatment for that. And then the last thing is there a standardized herbal product available which could be used? And again, FACA is a good example of that. So I don't need to repeat this because Sylvain has already presented it. So this is the example I'm going to be using throughout this talk. So the second step is to consider regulatory issues. I've seen there's been some chat about this already in the chat. Obviously, each country is different, but you will need to observe the national laws and regulations on clinical trials, for example, observing GCP. Do you have a suitable standardized herbal product? Is it licensed for use in your country? And does it need to be produced to GMP standards? Different countries have different rules about this. So I thought I would give you an example from the UK, which is a plant called Pelargonium, which actually is a South African plant. So we were able to do a feasibility clinical trial in the UK on a product which doesn't have GMP. But if we want to do a full-scale phase 3 clinical trial, we do need a product that has a GMP in order to do that in the UK. But different countries have different regulations, and we can discuss afterwards whether it's actually necessary to have full GMP. Maybe a standardized product actually is sufficient. So The next step is to really think about your research question in detail for the trial. And the system which is useful for doing this is what we call PICO. Thinking about who are the participants, what is the intervention, what is the comparison, and what is the outcome. So, oh, sorry, this was the pelargonium I wanted to show you. The slides were in the wrong place. So this is a South African herbal remedy, which is on the market in the UK, and it actually has GMP. There's a GMP preparation made by a German company called Schwaber. And we did a feasibility trial. We're currently preparing to do a full-scale clinical trial. Okay, so going back to the PICO question. So who are the participants? Thinking about FACA, should it be adults? Should it be children? Should it be both? And obviously we want to include people with sickle cell disease, but what should be the diagnostic criteria? We need to have a clear definition of what we mean by sickle cell disease. It could be people who are homozygous, heterozygous, both. You know, what diagnostic tests do they need to have had? Then the interventions. FACA comes as a capsule, it comes as a liquid. Which one do we want to use? What dose are we going to use? What frequency? For how long? All of these things need to be defined. And then compared to what? Do we want to compare it against placebo or against usual care or against a conventional treatment? And then what outcome do we want to measure? Do we want to look at prevention of sickle cell crises or treatment of crises, maybe looking at duration or severity of pain? So there could be many different research questions if we consider all the different variables for the different parts of the PICO question. So in the end, what we decided for FACA was patients aged over 12 with sickle cell disease would be the participants. The reason for that is that they wanted to look at the capsules as the intervention, and obviously it was felt that children under 12 may not be easily able to swallow the capsule. Compared to placebo, and the outcome, the primary outcome, was going to be frequency of vaso-occlusive crises over a period of 1 year. So I'm going to explain each of these and the reasons for these decisions in a little bit more detail now. So, so this is our research question in PICO format. In patients aged over 12 with sickle cell disease, how effective are daily FACA capsules compared to placebo for reducing the frequency of vaso-occlusive crises? This is how to formulate a research question for a clinical trial. So in more detail, thinking about the participants first, we need to define exactly what are the inclusion criteria. So like I've mentioned, we decided on the age above 12 because they would be able to swallow capsules. The sickle cell disease was defined as hemoglobin SS and hemoglobin SC. Obviously, they needed to have signed consent or signed assent for children. And they had to reside in the study area so that they would be able to return for follow-up. They would need to agree to return for follow-ups. And we wanted people who had had at least one crisis in the past year, because obviously if people are already well controlled, they don't have many crises, then it wouldn't be very useful to include them in the trial. And then we need to think about exclusion criteria. So obviously we want to exclude people who can't swallow capsules, those who are currently taking another effective treatment. So hydroxyurea is the conventional treatment, or someone who's already taking FACA. Obviously we wouldn't want to include them because we couldn't randomize them to receive a placebo. Like Sylvain mentioned, pregnant or breastfeeding women would be excluded because there's no safety data in that group. And then there was another list of patients to exclude: those with mental disorders, chronic renal failure or heart failure, known allergies to the study drug, that's an obvious one really, and people taking part in any other ongoing clinical studies, or someone who's currently having a severe crisis. Obviously wouldn't be appropriate to recruit them into the study. Now, in terms of the intervention, as I mentioned, the team decided to go for the FACA capsules being taken daily over a period of 1 year. Now, the control, it was decided to go for placebo. So you might ask, why placebo? So in this case, the standard of care in the national guidelines doesn't include any treatment for prophylaxis for prevention of sickle cell crises. So if the national guidelines recommended a certain treatment, then you can't really randomize people to placebo. So for example, someone with severe hypertension, it may not be considered ethical to give them a placebo if there's already an existing effective treatment which is recommended in the guidelines. So you have to consider whether it's ethical to offer a placebo. Obviously, if it is ethical, then a double-blind design can be used with a placebo, which is ideal because it helps to ensure that the reported outcomes are not affected by the knowledge of the treatment allocation. It helps to control for the placebo effect. But as I said, that may not be appropriate in all cases. So in terms of the outcomes, there are many possible outcomes, and this is a long list of all the outcomes that we discussed, but you do have to decide which is the primary outcome that you're going to choose, because, as you'll see in a minute, that determines how you calculate the sample size. So after discussion with the study team, we decided that the primary outcome we were going to choose was the number of sickle cell crises per year requiring a medical consultation. And all these other things are listed under secondary outcomes we were also interested in, but all of these are what we call secondary outcomes. So it means we're going to collect data on all of this, we're going to report all of this, but in terms of defining whether or not the product works, it would be determined by the primary outcome. Was there a significant difference in the number of sickle cell crises between the treatment group and the placebo group? And you'll see the secondary outcomes contain some outcomes about efficacy, number of crises that need NSAIDs, opiates, hospitalizations, length of time to the first crisis, duration and severity, and their hemoglobin, and use of analgesics and all-cause mortality. And we also had outcomes on safety, so incidence of adverse events and serious adverse events, laboratory parameters, full blood count, renal function, liver function, and side effects. So how do you decide which should be the primary outcome? So it's really important to do some community engagement and to find out what is the most important thing for patients. So in the UK now, our funders require us to do public and patient involvement prior to starting any clinical trial. And this is the sort of thing in which we need their input because we want to choose an outcome which is meaningful and most important for them. It has to be clearly defined and measurable. And for global health funding now, certainly from the UK funders, they also require there to be some community engagement in the process of preparing the proposal and in defining, for example, what we choose as the primary outcome measures. So then the next question is, what should be the sample size? So there are lots of things to consider, but the reasons that you want to calculate a sample size are to ensure that you have a good chance of finding a difference between your two groups, if there is one, and to avoid an erroneous conclusion. So it's possible to have incorrect results from a clinical trial. You can have a false positive result, which is what we call a type 1 error, which is when you find a difference when in reality there isn't one. So you come up with a result saying your product is effective when really your product isn't. And that's why we have the 5% level of statistical significance. And then you could also have a false negative trial, which is a type 2 error or beta. And that's the risk of not finding a difference when in fact there is one. And this is what we try to minimize as well. So the risk of this error, we typically try to keep it to less than 20%, or if possible, less than 10%. So 1 minus beta is what we call the power of a study to detect a difference. So normally we aim for 80% at least, 80% power. So in order to estimate the required sample size, you need to know firstly, what is the primary outcome measure? What is the expected value of this outcome measure in the control group? So without treatment, how many sickle cell crises do patients experience in a year? And to get this, you have to look at the literature. You have to find other similar studies and look at what they found. And then next, you have to think, well, what is the expected value in the intervention group? What is the minimum clinically important difference? So what would be a meaningful reduction in the number of sickle cell crises for patients with sickle cell anemia? And again, for this, community engagement, patient and public involvement is really important. What would be a meaningful improvement for the patients from their point of view? And then you need to consider what statistical method are you going to use to analyze the results, because that determines how you calculate the sample size. I have to give you a caveat. I'm not a statistician, but if you have any questions about this, I think that David Petrov is also on the call and he can answer all your statistical questions because he did this part of the proposal. So the example of the FACA trial, the primary outcome measure is the number of sickle cell crises in 1 year. The expected value in the control group, we looked at the literature and we found a recent trial which was looking at another treatment versus placebo, and we were interested in the placebo group. So the median number of crises per year in this study was 3, or 2.98, but let's say three crises per year. And then there's a Cochrane review on hydroxyurea versus placebo, and they found annual rates of up to 7.9, so say 8 crises. But being conservative, we assumed 3 crises per year, and then we aimed for a 2/3 reduction, in other words, reducing from 3 crises to 1 crisis per year. And in terms of the analysis, the statistical team decided to go for survival analysis, Kaplan-Meier plots and Cox proportional hazards model, comparing the onset of the first pain event in the groups and adjusting by the history of crises in the previous year and using intention to treat analysis. So this is actually quite a complicated method of analysis, and it's not the easiest for which to demonstrate a sample size calculation. So I'm not going to show you the calculation. To calculate a sample size, basically you need a statistician, and the formula depends on your outcome measure and your planned analysis type. If you have a binary outcome measure, like, you know, someone's positive or negative, someone's either cured or not cured, that's fairly easy to do. There are some standard formulae that you can use. But for survival analysis, it's more complicated. You really do need a statistician, but essentially it's done using statistical software like Stata. So in the example of this study, the plan was to use Poisson regression, and it would have 85% power at a significance level of 5% with a sample size of 81 per group. If you want to know how the statisticians came up with that, don't ask me, ask David. And then we increased it for up to 100 patients per group to take into account dropout. It's not unusual for people to drop out, so normally we add about 20% or 10% to the sample size to allow for patients who drop out. So the sample size was 200 patients. You need to consider other things in determining your sample size. How much budget do you have available? What's it going to cost? How many study sites would you need? And how feasible is it to recruit? How many patients do you see per year who would fulfill the inclusion criteria? And of course, not 100% are going to agree to participate. So you have to estimate how many you think are likely to take part. And then you need to develop a budget, thinking about all the activities and the associated costs. What does the intervention, the treatment cost? What does a placebo cost? Often that can be quite expensive if you're having it made to pharmaceutical standards, to GMP. How many visits per patient? What needs to be done at each visit? How much clinician time? How many blood tests, including travel costs for the patients, compensating for their time, and allowing for emergency treatments if needed. What happens if the patient needs to be admitted to hospital? Will the study pay? And I think it's important to think from an ethical point of view as well. What medical care would you want if you were a participant? You would probably want there to be a competent clinician on site at all times, regular follow-up visits, someone who comes to visit you at home if you're not well enough to go to the health center, and to know that you can go back anytime in case of any problems and given a second line treatment if you're not improving, and emergency treatments if you become seriously ill. So all of this can be quite expensive and it needs to be factored in. This is just an example of a study we did in the UK, flowchart showing the different steps to which people are randomized and the different follow-ups. So it's quite good to have a flowchart like this to help you think through what your trial is going to look like. And obviously think about other things in the budget: salary costs, a multidisciplinary team, database, software costs, community engagement and public involvement costs, ethical approval costs. And then once you've done all of that, prepare your funding proposal. You need to find an appropriate funder or call, get a multidisciplinary team together with the appropriate experience, write the proposal according to the guidelines. And the main thing is allow enough time. It takes a long time. And it's funny how often people contact me saying, well, would you be interested in taking part in this? The deadline is next week. And 1 week is just not enough time. You need plenty of time to develop a good proposal. So that's it. Good luck. And if you have any questions, I'd be happy to try and answer them. Thank you.
Oh, thank you so much, Merlin, for the excellent talk. It was really informative, detailed, and to my eyes, it's not only a guideline, but a roadmap how to design a clinical trial. Actually, with your questions, more questions are coming, but still it's so useful. And seeing the chat box, many questions come up. And if I would like to do an abstract of this, I would say the keywords are regulations, GMP, ethics, and budget. So these are the summary of questions, but let's allow 2 questions. Is anybody would like to do it, or we can ask from the chat box? Let me see. Yes, could you a bit discuss about GMP and the requirements, and especially in Africa, for example?
Well, I think Sylvain would be better to answer that one than me, to be honest. I'm not sure what the situation is in Burkina, but I've worked in Mali, and I know in Mali they don't require GMP. They have a system for standardized improved medicines. I believe the same is true in Burkina Faso. I think, to be honest, the GMP requirement, it's a real hindrance even to us in the UK to do clinical trials on herbal medicines. There are many herbal products on our market in the UK which don't have GMP, which people are using every day. So to my mind, I don't think we should need it, but we don't make up the rules. So we have to follow what our government decides. Yes, so in my opinion, you shouldn't have to have GMP. But, you know, at the end of the day, we have to follow whatever the national guidelines are and or the national laws. So maybe Sylvain can answer from the point of view of Burkina.
Sylvain?
Yes, Merlin.
Yeah, comment on it.
Yeah, we consider GMP, but we are very realistic, because we cannot do like a conventional medicine. Yeah, and so I think that together we can have a guideline for phytomedicine that to go to the trial, the clinical trial. A guideline, with rigorous guideline, but realistic too for us. Yeah.
Okay. And another question is about the difference between the first trial you mentioned and the final
feasibility and then a full-on trial. Yes. Yeah. So a feasibility trial, the aim is to answer the question, is it feasible to recruit and retain patients to a trial like this? So you're not answering questions about efficacy, you're not answering questions about safety. So there isn't a sample size calculation based on effectiveness. The trial is not powered to detect a difference between the groups. And therefore, our regulator, the MHRA in the UK, allows us to do a feasibility trial on a product that doesn't have GMP, because we can't make any claims about effectiveness at the end of it. All we can say is it was possible to recruit patients to a clinical trial of this product, and it was possible to follow them up. It's quite useful to have done a feasibility trial in terms of justifying further funding, and you may see a signal of effect. So you may see something of a difference between the two groups that may not be statistically significant. But we've done about 3 feasibility trials now at least, and it's very difficult to move on to a full-scale trial because you need that GMP product in the UK context. But in terms of numbers, which was David's question, the numbers are smaller. There isn't a specific formula for calculating a sample size of a feasibility trial, but it depends how many groups, but probably you would want to maybe have 20 patients per group or something like that. I mean, it depends a lot on what the study is and, you know, what condition you're treating and so on. Okay, and the question about how do you handle the ethical clearance?
Yes.
I mean, in the UK, we have to go through our ethical committees like for any other trial. We had to prepare an investigator's brochure with all the information we have on the herbal product, on efficacy, safety, and so on. Maybe Sylvain can answer for Burkina, because I wasn't involved in the ethical approval for or proposal for the trial in Burkina.
Maybe we can discuss about it more at the end. Thank you again, and for all your efforts, not only your today talk, but all the efforts all these years to promote, I would say, herbal medicine and the safety and preclinicals and all your work. Thank you again.
You're welcome. Thank you for inviting
and sorry,
me.
could the speakers maybe just respond in the chat box as well, because there are lots of questions there. So we can move on to the next speaker, but it would be good if we also get answers in the chat box. Thank you.
Thank you. So is Professor Matsabisa with us? Are you?
Yes, I am here. Thank you so much.
Can you share your screen and we can start with your talk?
Thank you so much.